Okay, people, this is going to be the remainder of the lecture that we didn't get to today, that I was storming through, but we just need to spend a little more time on it, and this will make it a little easier for you to fill out the packet as well. So we left off, we were talking about hormones and what they do, particular hormones with particular effects. So when we look at the two hormones, the luteinizing hormone and the follicle stimulating hormone, both of these are going to be gonadotropins. So affecting the gonads, the testes, and the ovaries. And then remember that tropins, we've got both tropic hormones and non-tropic hormones. So tropic meaning level, and so the tropic hormones are going to be the ones that cause other hormones to be released. So we see that luteinizing hormone targets the Leydig cells, causing testosterone to be released. So that makes it tropic because it sets up a level. Non-tropic hormones are the ones that go to the target cell and they do a specific action that is not endocrine in function. So when insulin goes to a body cell, it causes the body cell to absorb glucose. So that is non-tropic. All right, so our Leydig cells are going to be the target of luteinizing hormone. So we can remember L-luteinizing, Leydig, and these are going to secrete the testosterone to create the sperm. And then we have follicle stimulating hormone, S for Sertoli cells, to develop the sperm. And you can remember this because you have to make the sperm before you develop them. And so luteinizing hormone is going to come first. In the female reproductive system, we are going to have much more complicated processes. So our follicle stimulating hormone, that is going to mature the egg. So if we remember that our primary oocyte that was developed in utero in the first few months of development. All of the egg cells undergo the first round of or the beginning of meiosis until they are in prophase 1. Then the follicle stimulating hormone causes those eggs to develop from prophase 1 to metaphase 2. But this also causes the developing egg cell to release estrogen, and that causes the endometrium to thicken. And then it also causes the release, and then the estrogen causes the release of luteinizing hormone. So the luteinizing hormone takes the cells around the egg, and that develops the follicle cells, and they release the, they keep developing until you have ovulation, and then those follicle cells develop into the corpus luteum, and that's going to release progesterone and maintain endometrium. It is unlikely on the AP test that you will have to know all of this information out of the top of your head. It is more likely that they will give you some type of chart, and you have to trace the feedback loops of the chart for these things, but it will be helpful to have a general knowledge of what works so that it makes reading the chart easier. All right, adrenaline and noradrenaline. So this is your fight or flight response. So you get scared, there's a bear chasing you, you have to give a presentation, something. So the pituitary gland is going to release adrenal corticotropic hormone that causes the release of the adrenaline. And so the different things that this causes opens up your breathing passages, so you can breathe easier, get that oxygen to your body. So this is also why adrenaline, which is known as epinephrine, which is also what is in the EpiPen. So if you have anaphylaxis because you are allergic to peanuts and your airways start to constrict, you take a shot from the EpiPen, which is adrenaline, that dilates the bronchioles so you do not suffocate. Glucogen gets converted to glucose for energy. That bumps up your blood sugar. It also bumps up your heart rate, constriction of the blood vessels. This is from the norepinephrine. This is going to raise your blood pressure. And that's going to help pump the blood and the sugar and the oxygen to your cells more effectively. And then your eyeballs open up so you can see all those terrible things coming at you. And again, your digestive system, all of that kind of goes more minimum because you want the blood flow away from your digestive system to your muscles so you can either fight or run away. All right, glucocorticoids, mineralocorticoids, these are in response to stress. Glucocorticoids, blood sugar. Mineralocorticoids basically increase salt. So this is going to raise your blood pressure. And this will raise your blood glucose level. So one of your mineralocorticoids that we know about, that is also ADH, your antidiuretic hormone, increased salt absorption so that you can maintain water. Long-term exposure to the mineralocorticoids and the glucocorticoids, especially cortisol, that can suppress your immune system, which is why you can get stressed. You can get stress sick or sick from when you're stressed. All right, plant hormones. Again, quickly, auxins and cytokinins, these are both for cell division. And they have the opposite effect. Auxins cause stems to grow tall, but roots to grow sideways. Auxins are what we used in our sunflower seed lab. So instead of the roots growing long with the auxins that inhibited the apical growth, cytokinins do the opposite thing. Gibberellins, apical growth, most important is going to be for fruit and seed development. So as seeds are developing, they secrete gibberellins, which cause the fruit to develop. That way, when the seeds are ready, the fruit is ready to attract the animals to eat them and spread the seeds. When the seeds get in the ground, gibberellins cause the breakdown of the starch into sugars to allow the plant embryo to grow. That is going to be blocked by abscisic acid, which is typically written out as ABA. And that is going to block cell activities. So this happens for seed dormancy. When it is very dry, ABA gets secreted to cause the stomata to close to block transpiration. And then ethylene gas, this is basically formed as tissues die and rot. And so that's basically what fruit ripening is. The tissues are breaking down, so the starches become sugars. The green color in the fruit breaks down, and the accessory pigments show up. And then that's what causes leaves to fall in autumn to prevent water loss during the drier winters. What you should expect on the AP test from this is information about these hormones, and then you have to apply them to certain situations. They may give you some feedback loops about different processes that you have to track. So again, it will be very helpful for you to be familiar what these do before you read those charts. Phytochromes, This is just a cell signaling response to different kinds of light. So remember your phytochromes, you've got PR. This absorbs red and gets converted into PFR. PFR absorbs far red light and then gets converted into red. So this is going to control seed germination. So you have to have, so certain seeds needs a little bit of light in order for the dormancy to break. And then you also have the shade avoidance response. So in the shade, you get more far red light. So the PFR gets converted into the red form, and then the red causes apical growth to get away from the shade. In bright light, you have more red light, which converts it to the far red, and then that form causes more branching outward growth. What you might expect for the AP test on this is you'll give a paragraph of information, and then you'd have to potentially apply it. Well, you actually probably won't have to recall this specific information. Things you might have to recall is phototropism, which is going to cause the growth of the plant towards the light. That is because on the side of the light, auxin production is inhibited. So your auxins are pumped over to this side. So these cells over here elongate, causing that to grow that way. There have been multiple free responses on this very idea. And typically what you will see is they'll show this, and then you will have different seedling. This is called a hypocotyl, which is the embryonic shoot. And so you'll have this where it is blocked with a cap, which means that the auxins won't be produced, and this will just continue to grow straight. And then you'll have some where you have it's been cut off, and then there is like some soaked chemical with auxin, or a soaked piece of like agar or cloth with auxin in it, and then you have to depict which side that it will grow based on the auxin concentration and just different situations like that. So just know that auxins cause cell elongation. So where there's sun, it's going to grow towards the sun because the auxins are on the shaded side because the light blocks the auxin. Gravitropism or geotropism, roots growing down, shoots growing up. That's because there are little starch granules in the cells. That is your stimulation. That's your cell receptor, that the movement of those granules. And then your cell response is different in roots. That causes the cells to grow down, but then shoots, they're going to grow up. And then you have thigmotropism, which is just the auxins on the outside are produced, and that causes them to wrap around stuff for support. Okay, so quorum sensing is one last topic that I think I meant to talk about at some point, but then I forgot. I meant to talk about it in ecology. So what quorum sensing talks about is when you have individual organisms, when they get into a high enough group number or concentration, their behavior will change from individual behavior to group behavior. So we see this in bees. So bees will go out. They will potentially be trying to find a new nest. One worker bee will say like, hey, this is a great new nest. She will fly back to the hive. She will get some of her sisters. They will fly. They will check out the area. If those sisters like it, they'll go back and get some more. And they'll keep doing this back and forth until enough of the group has approved the new nest. And then the nest will leave its old site and go to the new site. So that's a form of quorum sensing, kind of at the organismal level. Down at the cellular level with bacteria, what we see is you've got these individual bacteria that are just doing their thing. They're growing, reproducing, But they are also secreting these chemicals that are called inducers that essentially do nothing until there are enough of the bacteria that the concentration of the inducers is high enough. And when the concentration of the inducer is high enough, they bind to the cells, and then it changes the signal transduction pathway in that cell, causing them to do something else. So this is how you have glow-in-the-dark bacteria. When they are in a low concentration, the bacteria do not glow. But when they get to a high enough concentration, and there's a high enough concentration of these inducer molecules, that stimulates the bacteria to start glowing in the dark. This is also seen on your teeth. So you've got bacteria growing in your mouth all day long, and when they get to a high enough concentration, they start producing what is known as a biofilm. If you've ever kind of got to the middle of the day or later at night and your teeth feel fuzzy, that is the biofilm proteins that all of those bacteria have secreted, and that is a protective mechanism to protect them from your saliva, because then they will create their own microenvironment in that biofilm, and they will start secreting acids, and they will wear down the enamel and give you a cavity. So the brushing action of teeth breaks up the biofilm, and then most toothpastes have some form of baking soda in them to make the toothpaste basic, and so that it will neutralize those acids from those bacteria. So this video up here, if you go through My Baylor and you watch it, it's super interesting. It's one of my favorite videos about quorum sensing. It has Dr. Bonnie Bachelor, and she has done all kinds of work with this, and she talks about how they are trying to use this quorum sensing idea to develop new types of antibiotics, because what will happen when bacteria are individuals, there's no way for them to really hurt you, but when they get to a high enough concentration, then they start producing what are known as exotoxins, and that assaults your body and your immune system, and that's when you start feeling things like food poisoning. So really interesting video. It'll kind of talk about this more. It also talks about a super cool mutualism between these glow-in-the-dark bacteria and this squid, which is amazing. I hope that after we get done with all the review and we're in kind of AP review week and we need a break, we may watch this video because it's really good. But still worth your time. Okay, so that is it.